So when we perform microscopy, and this was kind of the last slide I had on my other presentation, we are using a very particular type of microscope to do this. So microorganisms have the same refractive index as water, which just means that when you put them in an aqueous solution and then try to shine a light through them, you're not going to see anything because they're clear. So we use a compound microscope that has an iris diaphragm on it. And what this little thing does, which you'll see in a minute when I put a slide on here, is it changes the angle of the light shining through the slide. So it casts a shadow, and that allows us to see the organisms and their outlines and some of the structures inside of them. And that way we can identify them. And if anyone wants a quick lesson and wants to observe their own sample and use the microscope, you can come up here and do that as well. Pull up a chair. I've got water. And I was going to offer it up to, we're not going to be able to look at all of these samples in this short amount of time we have, but I will have the microscope here, and I might be willing to set it up somewhere later so we can nerd out, because this is not actually the only microscope I own. I own another one and a pocket one and the tiniest microscope you've ever seen in the world, which I wear as a necklace. So, nerd. I am a nerd. Yes, it does. It was a graduation present from a friend, and it's freaking adorable. Yeah. So as you saw, we added a little bit of this compost sample. So the process I'm using is specifically for performing assessments. Biological assessments of microbial activity in compost is something I do as work. We have spreadsheets that perform calculations that can help predict how many microorganisms and the diversity based on just this one tiny sampling out of your compost. It's pretty exciting. If anyone is familiar with like ecological surveys and stuff, this is just that on a microscopic level. And we shake it up like this because we have to knock the microbes ideally off of the particles of material and the soil and get them out into the water so we can observe them a little bit better. Because it's hard to get chunky stuff underneath the cover slip. If you do come up here to hang out and look at your own sample, I will caution you, cover slips for microscope slides are these impossibly thin slices of glass. They disappear and end up in your foot very easily. So, you know, all the fun cautionary tales. Yeah, so microscopes actually are a pretty accessible way to be able to track the health of your own compost. This particular microscope is, in all reality, quite a fancy version. But we knew it was going to be being used for educational purposes, so having some slightly nicer equipment is can be a good thing. Most of the cost of this camera or microscope is in the camera. High quality cameras and software can be very expensive for microscopes. You can get much cheaper versions. Probably somewhere in the realm of like $200 to $300, you can get a perfectly acceptable microscope that you could use yourself for viewing samples. This one is a trinocular, so it has the two eyepieces plus the tube here. I need at least a binocular microscope because when I'm performing assessments for work, usually I'm doing multiple in one day, which takes several hours. And it can be really strenuous looking through a single eyepiece for long periods of time. But if you're just looking at your own compost samples, that can really bring down the cost of your equipment. Mine is a trinocular because I also want the camera port, but to be able to comfortably sit and watch this. So I can go through a lot of samples and you can all watch me do it at the same time. So this microscope was not cheap. but that doesn't have to be the case. I think my first microscope ever, it still had a camera, but I paid $500 for the whole setup, and half of that was the camera. So, all right. And then we got to see exciting things. Oh, not yet. All right, so if you want, I can announce whose compost this is. Typically, so I teach community microscope classes in my community up in Paradise. And it's usually my policy that to keep it very unbiased, we don't say whose sample it is we're looking at. Often also because people bring samples from local businesses, and it's never my intention to like put anyone on blast, you know, and I certainly don't want to make anyone here feel insecure about their compost either. So you're welcome to announce it if you see me using your sample, but otherwise I'm not going to say nothing if you don't.
I don't care. You can totally share. Can you announce, sir? Yeah.
Go ahead and announce.
Okay, so some of the things that you're seeing here. Whenever you see these like really jagged particles like this, sometimes they're clear, sometimes they're not. Those are just minerals. Often you're going to see that in really sandy places. Sand particles, sand granules can look like that. Really sharp edges. You're still not quite seeing the same thing as me. Wowza. Yeah, so this guy, actually both of these guys right here, these are amoebas. Wish I could focus. Yes.
Wake them up.
So testate amoebas are definitely shy, particularly when I just scooped them into a bottle and shook the crap out of them. But there are certain things that you'll notice that'll help you identify. This kind of teardrop shape is really one of the most common ones that I see. So you'll see they have this hard shell. So this stuff that you're seeing, this kind of blorbly stuff inside, that's the actual amoeba. So unlike naked amoebas, which are what you might be familiar with, where it's just kind of this little clear blob that moves around with its little pseudopods, testate amoebas create this shell around them because, right, they're living in the soil. There's a lot of things that are out to get them, so it's just this extra level of protection. And you can see the outer shell here, and one of the biggest indicators that it's a testate amoeba and not something else is you can see this fainter circle, which actually we have a perfect view here. That's this little opening. You're just seeing it from a different angle. So this one is turned sideways where you're seeing that little cutout, and the other one is flipped on its back where you can see the opening kind of through the shell and on the other side. And if we ever let this sit long enough, you would start to see the amoeba kind of poking out. I've got some really cool videos on my laptop that I can show you later if you want. Often, though, you won't see them coming out of their shells unless you let this slide sit around for a while. There are ways to create these little humidors for your slides because the only problem with letting a slide sit out for too long is it'll dry out, and then it's going to totally change this. That amoeba might just go completely dormant, and then you'll never see it moving around. So amoebas are a super good sign in your compost. They are predators, which means that you've achieved a nice high population of bacteria. So something else you're going to look for is diversity, right? So you have a lot of amoeba. Do you have a lot of different kinds of amoeba? Because a lot of them are going to have preferences as to what kind of other organisms they eat. Having diversity is important because it turns out a lot of microbes are really picky about what they eat. And I'm shaking the table. So that motion you're noticing was just me wiggling the table. But you see this general just kind of vibration happening in the background. So those are bacteria. Most bacteria just kind of wiggle. They don't have a lot of ability to move around, but they do still move as much as they can. But there are other types of bacteria that are flagellates. So when we say these things, I know there was a picture on one of my slides. But you're going to see different types of bacteria. Bacteria are going to be really hard for me to zoom in on in this particular environment because we're translating that image from the microscope to the computer to the projector and up onto the screen. But bacteria are going to have lots of different shapes to them. These different shapes are going to indicate if they are pathogens or if they're beneficial. So round, rods, filamentous are typically good guy bacteria. And then there are certain shapes that are not great bacteria. Spirochetes, Spirilla, Vibrio, things like this are typically going to be pathogens. We don't want to see high numbers of those. These spiral type bacteria I see a lot in like manures and things like that. It's a reason you want to really get your temperatures up. Some of those can even be human pathogens. So when we're performing microscopy, we're looking at morphology, which is the shape of these organisms. And because of that, we can tell, oh, is he coming out? Is he coming out? Is that him? Did we wake him up? Y'all were so excited to see him awake. I think he might be doing it. He is. So that is an amoeba creeping.
So is it just coming out of the shell to eat or just like...
Yep, to move around, to eat stuff, check it out.
And go back inside?
Typically, they would spend most of their time out of the shell, grazing and moving around. That's exciting, I think. Sometimes it can be hard to tell what they're doing.
Is it the whole body of it come outside the shell? Like a snail or something?
Usually not the whole thing, because it still has a little bit inside there holding on to the shell so it can tote it around. So yeah, it would be like a snail. All right, so we're going to keep looking around. And you'll notice a lot of similar shapes going on there. That looks like a really big one. And actually, that might actually be something else, honestly. So you will find things like eggs from different microarthropods, nematodes. Different organisms lay eggs in your soil and compost. These look like some more types of testate amoebas. So you're also going to notice microorganisms in different states. A lot of them are going to insist or go to sleep. So technically we don't count those. So this is actually much too large to be a bacteria. So remember, all of these little guys floating around, the little white ones that you see, those are going to be our bacteria. This is a piece of cellulose. So the way that the different units that make up cellulose form is they create these super tight spirals, and that material doesn't break down super easy, so often you see it left over in your compost.
In the upper right-hand corner right there, there's the, on that golden surface, we have some.
Yeah, I was looking at that too.
What is that?
I don't know. Well, with those sharp points that are going on, I mean, there's clearly some crystallization going on, so you will see a lot of that too. Different salts and things can form different crystal structures under certain conditions. So likely it's mineral.
What about that kind of bluish wiggly thing to the right, top right of it? Is that an embryo? Is that too small?
Right in the center now.
Yeah, in the center.
So that's going to be an aggregate. So you're going to notice, even up top here, this big clump right here. These are all indicators that your microbes have started to create. So like I was saying, this thing is going to be full of bacteria, big groups of them that have glued themselves onto these food sources so that they have somewhere safe and they have that guarantee of nutrition. You'll also notice the different colors in this aggregate. You see that brown color? Do you remember what that is that's starting to form? It's the same stuff that was in this test tube. So see that same color? So that means that your compost is starting to mature. These materials have been broken down to that super core structure, and it started to pull in other materials from your compost substrate and create these aggregates. So these micro aggregates eventually will start to form macro aggregates once you get them into your soil. You may have heard people talk about like crumb structure being that ideal texture in agricultural soils. These little blobs of glue are what are going to hold all that together and cause that to happen in your soil. And it opens it up for drainage and also creates water retention because water is attracted to these big molecules and held onto the surface of them. And it's also going to create pores and airspace in your soil and in your compost to keep those microbes happy. The yellow is fulvic acid, which is a slightly less mature compound, but also an end product of all of this microbial activity. I wish I could get my gosh darn camera more in focus here. It's another testate amoeba.
Oh, there you can see it's opening from a different angle,
Yeah.
where it looks like a big round mouth, right?
Uh-huh.
Okay.
Yep, that circle in the end there. So typically for nematodes, we do a quick scan at a much lower power because nematodes are quite large and you can spot them pretty easily. I was going to see if I could find some boatloads of amoebas though, so good job, whoever's compost this is.
Is that red one is contamination, some kind of, there is a red colored piece, reddish,
Oh, that?
yeah.
No, probably just some kind of organic matter. It can be really challenging to distinguish every single bit of like food waste and stuff that's in your compost. So a lot of what we're looking for when we're looking for microorganisms is these very smooth surfaces. There aren't a lot of materials as feedstocks that you're going to put into your compost that are going to be smooth and rounded. So that's a really good indication that you're looking at living things. Oh, here, let's look at that guy. So that there down in the left corner. Oh, wish you had better resolution here.
So
I wish I
given
was
that
better
this
with
has been
cameras.
sitting for 2 days, are there things that have died off possibly in the sample?
Unlikely that they died. Most likely they went to sleep. It is possible, but also that container has a lot of airspace in it. So likely, since it's just been hanging out, organisms have probably had a chance to wake back up, and I just made them all angry by shaking the crap out of them. So I want to find a better, this one is particularly dark. So mushroom spores, fungal spores, this little seed-shaped thing, it's hard to tell on this screen, but it's actually very dark brown, and that is an indication of a fungal spore. I'm trying to find a slightly more see-through one. Tons of amoeba, just lots. And lots of different shapes of them too. Yeah, so a lot of, we found a nematode. He is not moving. He may not have survived. But they also do nap, so, you know, as we all should. Yeah, so the mouth parts are what we look at when we're assessing nematodes. And actually, this might be a good opportunity for me to adjust this camera a little better. So there are lots of different types of nematodes that we end up with in soil. Bacterial feeders are the most common because, again, their food source is bacteria. Ah, see? He's alive! They nap. I told you. I wasn't lying. They do nap. And yeah, so the mouth parts are what we look at when we're trying to distinguish different types of nematodes. And it's kind of hard to see. You see that bit of a flange on the end of its head, those big fabulous lips that it's got? Usually that is a good indication that it is a bacterial feeding nematode. I've seen some that have some structures that it's a little hard to believe are lips, but they're pretty fabulous. And you're also going to see this big area right here, that big wide opening for the mouth. That's also an indication that it's a bacterial feeder. So they're just sucking in all the bacteria they can as fast as they can. Nematodes are going to be one of the primary cyclers of nitrogen in your soil. That huge organism, they can eat thousands and thousands of bacteria every single day. And every time they excrete after doing that, they're releasing nitrogen back into your soil. There's a really fun method of measuring nematode diversity in your soil. You can actually extract them and then look at whole slides that are nothing but nematodes. It's a lot of fun. They're definitely the most exciting thing you find in soil, I'm not gonna lie. I show people like fungal strands and stuff and they're like, eh. And then we get a nematode and everyone's like, holy crap. And it is quite large too. And actually,
The inside of the nematode, is that, what are we looking at? Is that just is there like organs or?
yeah, so nematodes are not single-celled organisms. They have multiple cells, organelles, and things like that. There are different bulbs that are part of the digestive tract, which is another way that we identify different functional groups of nematodes. So you see there's a bulb here and a bulb here with that little T shape in it. Those are all part of their digestive and excretory tracts. Yeah, and you can see I zoomed out quite a bit and we can still see this whole nematode, so they're quite large. And we can skirt around. I'm going to stay at this higher magnification just for the sake of time. So this is usually what happens is we get to one sample and there's just so much that often we don't have a chance to move on. But we have time the rest of the weekend. That one is a mover. Someone's hungry. Oh, you can't see it. Let me see. There we go. There you are. So typically nematodes move very fast and are extremely hard to track. So I guess the last main point I wanted to make is there is a reason that we go through these pains to observe microorganisms live. And I'm not sure how familiar you all are with kind of traditional microbiological practices. Anyone who's taken a microbiology class, you know, you take your sample and you're trying to isolate a certain microbe and then you plate it out in these different nutrient medias, and then you put it in an incubator and you try to grow it, right? And then you count how many of them are still alive and reproducing. So the challenge with these types of methods, and also with doing things like DNA testing in a soil environment, are it doesn't actually tell you what's alive and metabolizing in that environment. So with DNA sampling, there may be remnants of microbial DNA from months and months ago. Those organisms may have long since died. It's also going to sample DNA from microbes that are dormant, which is great that they're still there, but they're not contributing anything to the soil environment, and there's no guarantee that they'll ever wake back up. So it's not really helpful in determining like what nutrient availability is going to be there and what kind of benefit these are going to pose to the environment and to our plants and gardens. So that's one issue. And then with traditional microbiological techniques, the problem is isolation. So none of these systems we talk about exist in isolation. It's one of the big problems with agriculture. We can't just take that nitrate nitrogen and add it back into the soil and think that we have a healthy functioning system. It's going to cause problems in the long run. It is stripping plants of nutrients, which in turn are not getting passed on to our bodies. So you can't just take one piece of the puzzle and think you're going to build this huge, beautiful picture out of it. And it's the same with microbiology. There are significant human pathogens that we are still unable to culture in a lab setting, and because of that, we haven't been able to find cures for very significant diseases. And it's the same with the soil environment. We can't mimic this complex environment in a lab setting. We're trying, but it's just not the same. Every tiny pocket in your soil environment is going to be completely different at any given time. It's going to have a unique moisture content. It's going to have a unique EC and pH. It's going to have a unique diversity of microorganisms and plant interactions happening. It's one of the reasons it's so hard to sample soil and get an accurate picture of what you're seeing, because you can't just take all these different samples, mix them together, and be like, I have an answer for what's happening. Like, it just doesn't work that way. These are very dynamic systems, and we have to be able to just take that snapshot, in this case in the form of a soil sample, and just look at what's happening. It's also one of the reasons that I don't have people send me samples to do this kind of analysis. I don't personally feel like that's an accurate representation of what's happening in your soil anymore, versus someone literally taking the sample, putting it directly onto a slide. And even then, you know, there's a level of manipulation which is going to affect what's happening. As we saw with the amoeba, like they sucked into their shells, and now I'm not 100% sure whether or not they're still alive. So am I going to count that towards your nutrient cycling? So lots of challenges, and it's one of the benefits to microscopy that we don't get with other methods of microbial identification. Where's your face? No. Where's your face? That's your booty. Wrong end. Does anybody have questions while I try to?
So with microscopy, we are just figuring out what kind of microorganisms existing in our soil.
Yeah, so the things that I would put on a report if I were doing this for somebody is we have all of the different functional groups. I need to see a certain number of organisms from each of those groups that I mentioned. So a certain number of bacteria, a certain number of protozoa, a certain number of nematodes for you to essentially pass this test and have a healthy enough environment that you are cycling nutrients at a rate that will support plants if you put your compost into the soil. I also supply a fungal to bacterial ratio, which can help you determine what the best applications for that compost would be, whether you have an agroforestry system or whether you have intensive annual vegetable production, things like that. We scan for pathogenic organisms. So I talked a little bit about pathogenic bacteria, but there are also fungi, things like mildews and things like that, that we can identify morphologically under the microscope, and we have to have below a certain threshold of numbers of those organisms for you to be able to pass. Yeah, and these counts are also contributing to just the great determination of diversity in your compost. Something else that a lot of people include in these surveys is nitrogen cycling potential. And what that tells you is, based on the number of organisms and the rate at which they are feeding on each other, what is the rate of production of plant available nitrogen that could happen in your soil by applying these composts. So that's a really pretty fantastic thing to be able to predict just with microscopy, and valuable if you're using this information to advise farmers and gardeners too. Mm-hmm. Good, because I was done.
Aside from compost, they reserved in the salt sea, they have, I think it's UC Riverside Organic Farm. They're doing cover cropping to fix the nitrogen.
Yeah, cover crops have lots of benefits, especially in an environment like yours. They're going to help hold things in the soil. They're going to also maintain the relationships with the microbes, because when you let your field go fallow and these organisms that rely on those plant roots in the soil to survive are gone, you're killing your biology. Certain things will go dormant, but certain things also will die. Fallow's not great. Cover crops too, being rolled down, can suppress weed seed germination because there's living plants in the ground that are taking up those ecological niches. Plants affect water dynamics in your soil, so having that activity is going to help you manage water. Yeah, all the things. Cover the soil.
Don't leave it naked.
Don't leave it naked.
So once I'm done with my tomato, rather than pulling it out, it's better to snip it at the lower level and leave the root for the microbes to feed.
Yeah, and that has a lot of other ecological benefits too. Yeah, we gotta go.